Welcome to our Upper Maumee Watershed speaker series. My name is Caitlin Smith. I did the last speaker series on diatoms, and I have a few notes to go over before we get started. So for everybody on Zoom, please make sure that your mics and cameras are off and you're muted. This is going to be recorded. If you have any questions or comments, we're going to leave those till the end of the presentation, and then we'll get to them. For those of you in person, hold your questions to the end. So to begin, Julie Davis Good joined Allen County, Indiana's Soil and Water Conservation District in May 2024, leaving the Indiana Institute of Technology as a professor of biology after over 25 years in research and teaching. Julie's roots in conservation came from a family of organic gardeners and floral producers. As a first-generation college student, Julie attended Hanover College and earned a PhD in cellular and molecular biology at the University of Pennsylvania. She loves to help people adopt effective practices, discuss outcomes, and make changes for environmental well-being. Her visit with the Upper Maumee Watershed Partnership will discuss water sampling data in the Upper Maumee Watershed over the past decades, focusing on nutrients and sediment levels in draining streams and ditches. And so with that, I'll pass it over to our presenter today, Julie Good.
Kat, thank you so much. As we get started, I want to introduce my co-presenter today, Gabe Curtis. Gabe has his master's from Purdue Fort Wayne in Dr. Bob Gillespie's lab just a few years back, and has been with the Soil and Water Conservation District in Allen County, Indiana for about 3 years. Yes. So Gabe is our expert today. I'm just the master of ceremonies. But more importantly, sorry, Gabe, not more important than you, but we are so grateful to be here. Our key today is connection and discussion. And don't tell Caitlin I said this, but you are welcome to break in with questions. And so if you see chats come in, Jennifer, you shout them out at us. Being a professor for a long time, I feel like the only thing that we do together, we'll do with a foundation of understanding. And so, please ask questions. And you can even shout if you're in the room, okay, for our masses that are here. Okay, so the first thing we got to do is to say thank you. And so, I honestly believe, certainly I could not be standing here, but I think Gabe will agree with me that the people on this slide have been incredibly important. A lot of the data you'll see today, almost all of it, I believe, has been processed through the Fort Wayne, Indiana City Utilities Office with professional engineer Anne-Marie Schmerchik running and overseeing that process that's in multiple labs. As I mentioned, Gabe came from Bob Gillespie, Dr. Robert Gillespie's lab at Purdue Fort Wayne, and so that was the Department of Biology and the Environmental Resources Center, which he helped to found with Bruce Kingsbury. We also want to thank the St. Joe River Watershed Initiative. A number of those folks, I think, are involved in the Upper Maumee Watershed program partnership. Sorry, Jen. And then the Maumee Watershed Alliance was one of those groups that Greg Lake and numerous others have worked with and continue to work with to do the good work.
And
yeah.
If I could just add here, not only do we have so much lateral support in our partnerships each year, but also the project that we are continuing today is just 1 year younger than myself. So it's been going on for a long time. So not only do we want to thank those who are supporting us year in and year out, but also our predecessors who had the vision and saw the importance of this incredible work. What we'll be focusing on today is our most recent chapter in that, but certainly to the future. And we'd like to start off with just that gratitude, just that we're just here carrying the baton the next step further, but there's so many that have come before us that have begun this work, and we hope to just keep it going and keep things moving.
Yeah,
so
absolutely. And as you can see at the bottom of the slide here, our county, Allen County, Indiana, Soil and Water Conservation District, the supervisors, the Board of Supervisors has overseen and permitted the funding to go toward these projects we'll talk about today. And I hope online is one of your members, Don Reckeweg, who maintains associate supervisor status, and we're so grateful for Don. So we also have to say thank you to the people who did the work. And so I think we're frozen. There we go. So I've already introduced Gabe. He is a watershed project manager at the district. And for this purposes, he has taken almost every sample you'll see today, because the data we're going to show is 2024. He has quality controlled all of the data, all of the samples, and those are tens of thousands of numbers. And then he led the group the past 3 weeks, I think, that have analyzed the data. And those names are listed here. Maya Rizzo is one of our program assistants. She's a 2022 graduate of Western Michigan University with double majors in hydrogeology and environmental science and sustainability. Jason Hartenstein is a current senior, sorry about that, Jason, a senior at Indiana Tech. He is a computer engineering major. He'll graduate in May. And then Camille Sherbowitz,
Yes.
okay, is a graduate of Indiana University, and she is currently a Resilient Indiana Technical Assistant. And so we call that the RITA program. Phenomenal opportunity for, I think, Defiant students to also think about going and working up to a 4 or 5 year contract with the state of Indiana to learn all sorts of conservation practices and to be stationed in multiple counties. The other person that I cannot fail to show my gratefulness is Greg Lake, the prior director of Allen County Soil and Water Conservation District. He led all of the years of strategy, funding, execution of the historical data collection that Gabe referred a moment ago. He deserves all the credit. Greg just did incredible things, incredible things. So one more level of appreciation, and that is to the caretakers of the waters that we are discussing. The Miami people, which of course, if you didn't know, is a mispronunciation of Maumee, which is where the Miami people got their name. The Odawa people and the Wayanda. And these are depicted beautifully in a summer undergraduate research project that I found from Defiance College from 2 summers ago, I guess. Unbelievable website here that you see on the history of the Maumee River Valley and history of colonialism and all the rest of it, but a beautiful description here of the Native American settlements. And the best description that I could find for the reason that we must honor the original caretakers of this land is taken from actually the Toledo Museum of Art website. And they say, it is our responsibility and aim to understand and continually share knowledge about the complicated history of colonialism in the space we now occupy, the peoples who were here before us, and the enduring presence and diversity of indigenous peoples in our region. They go on to say, we commit to building meaningful relationships with the indigenous communities and working to dismantle the systemic invisibility of indigenous people. And here's the part that applies to us really clearly today. Through shared knowledge and exchange, we will continue to learn and enrich this history as we come to know more. And that's why we're here today. So let's go. Our purpose today is to further our collaborations between Allen County Soil and Water and the City of Defiance, the Defiance County Soil and Water, and everybody in between, farmers, partners, watershed groups, all of us, and to answer our shared questions. And so already the City of Defiance and CAP, Conservation Action Project, and a whole bunch of other stakeholders, we've already had an active collaboration on an enormous grant effort that we made just prior to the holidays. So we're going to continue those collaborations. But the other big issue we have is source water protection. And so one of the sampling areas that Gabe will present today is the source water for Fort Wayne, which is the county seat of Allen County, Indiana. The source water area that feeds Defiance, of course, is the Maumee. And so we want to track that water and we want to identify problems and areas and sources of pollutants. And so that's what we hope to talk with you about today. So that's what we want to do today. Now, where are we doing this? And I have to thank Jennifer and your whole group here for this amazing map. We've tried so hard to find a beautiful map, and it's yours. This is just the most gorgeous map we could find. And so shown here are all of the tributaries into the Western Lake Erie Basin from the Maumee standpoint, the Maumee River Basin. And so, of course, in blue, light blue, you see the Maumee, get over here, here we go, the Maumee in blue, so the lower Maumee that leads right to the mouth in Toledo to Lake Erie. The area where we're working today is between Fort Wayne and Defiance, which is what we call the Upper Maumee in dark blue. Now, feeding into the Upper Maumee and finding its confluence in Fort Wayne is the St. Joseph from Ohio, which is different than the St. Joe, Michigan, which goes this direction up here. So we have to be really clear. There are two St. Joes, which have their headwaters, what, like feet apart, something like that.
Right.
Yeah. And the St. Mary's, which comes down through from Ohio again, Adams County, Indiana, and into Allen County, our county. So this confluence of three rivers leads to the Maumee. Now, what we will not talk much about today is the Auglaize River Basin, or the Tiffin, or the Blanchard. So what we're going to do today is show, and this is a great place to show this, if we zoom in a little bit, we will be showing data from a sampling site here on the St. Joe at the state line, here on the Maumee at the state line, and here at the St. Mary's.
Yes.
Okay, cool. So now,
yeah, so just to be clear on these sampling sites, the significance here is important. By collecting at the state line on the St. Mary's and the St. Joseph River, water flow is coming into the state of Indiana. So we're catching things as they are coming in. And as we monitor things on the Maumee River as it leaves the state, that gives us an idea of what is coming into Ohio, what's coming downstream, headed directly towards Defiance, which of course the significance here is incredible considering this is your drinking water source here. So,
and Gabe's point is important also because in order to show the data that Gabe and the team, Maya, Camille, and Jason have crunched the past few weeks, our sampling sites had to be put with USGS auto samplers that detect flow. And so we'll get into this a little bit more, but Gabe picked these sites specifically so we could have actual flow data and then be able to calculate how many pounds per day of nutrients and sediment is being dumped. So this is critically important.
yes.
Okay, so how do we do this? We do catch sampling. And that's shown here in an article from 2020 on the Indiana Prairie Farmer with two of the Soil and Water Conservation Watershed project managers. So Gabe's precursors here is Brooke Rudisell on the left and Joelle Beal Neff on the right. And they are standing on a river bridge. And that little contraption you see hanging down is essentially PVC pipes with dark glass bottles in them. And so if you haven't seen this before, what happens is they drop it down the river. Some of them are 20, 30 feet high, and you wait till they submerge, and then you pull up the samples and then transfer them into the storage containers to take to the labs. This is the only kind of sampling that we do presently at this many sites. And it is not automatic. It is not mechanized, in contrast to those USGS auto samplers I was talking about a minute ago. And so what happens next then is that we send it to the lab for testing. Now, some testing could be done on site. We're discussing that now because of funding cuts and potential barriers to get continuous data. But more quantitative testing is definitely done by our partners with City of Fort Wayne Utilities. And so we look at physiochemical, nutrients or other analytes, and biological indicators. And so a bit more information about the physiochemical, it's going to be temperature, dissolved oxygen. They'll take the pH, which is the acid-base range, conductivity and turbidity, meaning how much stuff is floating in the water sample. From the nutrient side, We will look at total suspended solids, and that's going to be one of the graphs that you see today, TSS as we call it. And then nutrients will be the other graphs that Gabe has compiled, which is going to be phosphorus, nitrogen, which is a combination of nitrate and nitrite, and then ammonia. We can go further and do atrazine testing, and I believe we have those data, but we did not crunch those numbers for today's purposes. Mm-hmm. And then the biological surveys are done for coliforms, could be macroinvertebrates. E. coli is one of the coliforms that we want to see. So just to give you a sense, inside this tiny little box over here is a hydrogen proton. And it's that gives us the acid base, right? The logarithmic scale of pH is the proportion essentially of hydrogen, which is tiny, tiny in there. Now, this is a molecule of atrazine. So each one of those hydrogen atoms is there, the little white guys. And if we were going to look at the size of E. coli, it would be at least the size of this room that we're in, which we're in a classroom at Defiance College. So thinking about the size difference of the things we're detecting, it's super powerful, but I think we can all see why it's needed that we do them in a laboratory quality control manner. Mm-hmm. Okay, so that's what we do. Now, where do we do it? And so thanks to Maya Rizzo, one of our program assistants, for developing this map through an OpenStreetMap application, which maybe some of the students have used before. She has compiled the most amazing view of what Greg Lake and the Allen County Soil and Water, in partnership with all those people we mentioned on the first slide, have conducted over the past, gosh, Gabe, 20, 30 years.
Right. At least since 1996 that I'm aware of. Yep.
Okay, it's craziness.
Yeah.
Okay, it's wonderful. But on this map, Maya has coded the historical sites, meaning the sites that were prior to 2024 in brown. So all of the brown dots here you can see are places that we have data long-term. So through the St. Joe, and over here is Cedar Creek watershed. You can see the brown dots were historical data down on the St. Mary's, as well as historical sites through the Upper Maumee from Fort Wayne, and I put the star on Defiance since that's where we are today. Now, more importantly to what we'll be looking at today, this map is going to be the color scheme that we're going to follow through the day. So the Auglaize I mentioned, we don't do much there, namely because here's the state line. And so just a small portion of the Auglaize is in Indiana. And we do have one current sampling site on the Flat Rock Creek, which is in the Auglaize drainage basin. And that flows, as you Defiance people know, right into the Defiance area as well, downstream of your source water, if I'm recalling right. Yes. Thank you. But all of the blue dots are going to be dots that we have data on from 2024. And as I mentioned, Gabe has centered the work. So if we zoom in here, we can see a little bit easier the blue dots. And Gabe has centered our discussion for today on these three areas. So in the blue St. Joe, we're right here on the state line at Indiana and Ohio, but it's the St. Joe Basin. Down here is the St. Mary's. It'll be red in all of our other maps. And so it's again at Pleasant Mills, Indiana, but it's right there at the state line. And then up here, we've got an Indiana sampling site at the Upper Maumee, and our USGS partner that we found is just into Ohio in Antwerp, if I'm remembering.
Correct, yeah.
Mm-hmm.
But because those stations are relatively so close to each other, when you consider the whole watershed, the flow data there is going to be comparable enough that we could utilize it. I just want to touch on that again. I know Julie mentioned it before, but the significance of because when we collect our data, what we get back from the labs is concentration. So to be able to combine those concentrations with the knowledge of what's the volume of water going through that point at that moment, that allows us to then, jump up a mountain and be able to say, hey, now we know how many pounds of this nutrient is going through here. And what we did, as you look at our data today, what we did is we essentially applied that concentration and that flow rate for a 24-hour period, and that's how we did our math. Everything that you will be, when we talk about pounds, we're talking pounds per day. How many pounds of that nutrient, or TSS in that case, the amount of soil that's moving through that point in a 24-hour period. So that's what you'll be seeing here in the following.
That's fantastic. So what we're going to do is get started with the river we all share. So we're going to spend the most time right here in this yellow basin, the Upper Maumee River Basin. We're going to focus on this sampling site. We have a ring in yellow, and we will be showing 4 graphs to start with. And you know, if you want to get a cup of coffee and you hate numbers, now would be the time to do it. But we're going to walk you through just a little bit of data so that you see the power of what is possible with the sampling that has been done by this group, this partnership over in Allen County. So we're going to look at 3 nutrients. We're going to look at phosphorus, as I mentioned, total nitrogen, which is the nitrate and nitrite, and ammonia. All right, as well as a graph of total suspended solids, which I mentioned a moment ago. Each graph is going to be paired with weather events. So precipitation is going to be on the left side of your screen because it matters. And this is what's amazing is we've started this with nutrient excess is spring rain driven. And so we've got this beautiful way to show you the coordination of rain with nutrient loss into the rivers. Okay? All right, so here we go with the Maumee River in this yellow watershed. So the first graph here is at the Maumee River near the Ohio-Indiana state line. Along the left side, you see that we're going to have our nutrient load in a linear axis. And across the bottom are the sampling dates. And just to make things simpler, we've only put the months here, but note that there are 4 points for every month because Gabe does his sampling every Tuesday religiously. Yeah. And that, I should say, the entire route is about 100 miles, I
think more like 220.
220. Yeah. No wonder the mileage is so
yeah.
high. Okay, sorry about that, Gabe. So we have these data from every Tuesday throughout the time from the 1st of April. I guess we started on April 9th and went through middle of October. Off to the right side now, so the phosphorus, in this case, the nutrient is going to be in orange. And on the right side, the precipitation at this point on the Maumee never exceeded 1 inch, and it's shown in blue. All right, Gabe, yes. Take it away.
Yes, just to add to the precipitation, what we're looking at with these numbers is the amount of inches of rain that fell in the 72 hours that led up to us being at that stream. So this is it's a 3-day total of an average. We took multiple rain gauges across the watershed, averaged them together, and that's how we got those values. So I want to work left to right today. We're going to start with this precipitation point right here this first week. You'll see this drastic difference between the amount of rainfall and the amount of phosphorus that we had coming down the stream. And what's not shown on this visual that I want to make you aware of is when I went back one more week on the precipitation to look at the amount of rainfall that we had, the spike of blue would be twice as tall as our orange dot here. It was roughly about just over 2 inches of rain that fell the week prior. So I think what we're seeing here, this high amount of phosphorus that on our first week was coming down the stream. What we're seeing is we were still coming down from a rainfall that had ended 3 or 4 days prior, or 3 days prior to when we start calculating precipitation. So just that it started, basically the rain started on April 2nd and ended on April 5th. So it didn't show up on our 72-hour control, but really it's there. So we're seeing that. And what I'd like to draw your attention to is also the space under the curve here for April and May. So one of the things we came into this year's data knowing from last year's analysis was that there was a large amount of the nutrients that came down the stream came down during those April and May time periods. So what I want to draw your attention to is this area under the curve here. And then notice the rain keeps going, but the response in our nutrients diminishes until what we'll keep track of is just after the first week of July here, we see a little bit of a spike again. And then even though the rain keeps coming, we are seeing our nutrients sort of calm down or flatten out. So that was phosphorus. Now let's keep going with nitrogen.
And the same axis orientation. Left is the amount of nutrient up, and as Gabe already said, that high spike on April 9th is likely coordinated with the prior week's rain. And the rain again, the precipitation is in blue.
Yes. And so working left to right again, we're seeing sort of a very heavy nutrient loading in the first 2 months of our sampling window. And I would say just generally, I'm seeing more of a response with the nitrogen. And from what I've learned in the past few years, just attending other events and hearing other presentations on water quality, that the nutrients in nitrogen, they have less of a tendency to be able to latch on to the soil particles. So when it rains, it makes sense from what I've learned that we're seeing more of a response.
Soluble.
Yes.
It's solubilized in the rain.
Coming in the rain. And so then let's also notice here again that right after mid-July or just towards the front end of July, we're seeing a response again to the rain, even more dramatic than the total phosphorus. And then the same trend for the rest of the year, we're seeing that nutrient loading kind of level out or diminish. So that was nitrogen. And then we also wanted to look at ammonia.
Yeah, and this one does blow me away, Gabe. I mean, I was not expecting to see this. And again, so we have the same kind of area under the curve that Gabe been talking about from April 9th to about the 1st of June, end of May for sure. And then this ginormous, sorry, that's not scientific, this extremely high spike at the first week or two of July. So so that is extremely surprising, but ammonia. So we've got one more indicator to look at, Gabe.
So, and I think, so originally this would be TSS, correct?
Total suspended solids.
So we're looking at total suspended solids here. And originally we didn't have this in our analysis, but really what led us to ask the question of what TSS did was that spike in mid-July. That sort of surprised us, and that prompted us to ask the question of why are we seeing a spike in mid-July? And I think in the past, the title of our presentation, we were expecting to just be examining the spike in nutrients that happen in April and May, that the spring loading rates that are spring driven. And when we saw that spike in mid-July, it sort of raised the question of, okay, what really is causing the pattern here? And after thinking about it more, we started to realize that maybe nutrient application was more of a driving force than our necessarily just our spring rains were. Because certainly the rains keep coming all year, but we saw that diminish in the nutrient response. And so after the way we thought we could maybe test that is, let's check out something that's not a nutrient, but that we can also calculate loading rates with. And that would be the total amount of suspended solids. And sure enough, we don't see that same spike in mid-July that we saw with our nutrients.
And so Gabe and I were bantering back and forth for the past week, and we think about it all night, and we come in the morning and think about it again. So encouragement to all you students who think about things in the dorm room at night or after you've graduated, right? And so what hit us with this is Gabe was like, phosphorus should be bound to it. This should have phosphorus in it. Why is there no sediment? And it hit us that by the 1st of July, even though we had a very late planting season this year in agriculture, lawn care groups, golf courses, everyone's applying extra nutrients. Yes, the farmers do their nitrogen application, their phosphorus application in late, and we found out this year from Greg Lake, actually in late
late June, and they ended in the first week of July.
But the plants have roots. And so this actually goes to the point of when there are living roots in the ground, which is one of the 4 principles of soil health. When there are roots in the ground, it holds the sediment. And, you know, we'll have to do some more hunting through our data to see what else we could learn from this. But at the top of this graph, you see the arrows. So I just tried to indicate the rain events. And obviously we put that one off the left side of the axis here, so you can see where that huge rain event was prior to the first sampling we did. And then in our next data graph, we're going to put all 3 nutrients together. We're going to leave total suspended solids out. As Gabe mentioned, it's not a nutrient. And so because the graph range is so different on those graphs we've already shown you, we had to put our nutrient left axis on a logarithmic scale to be able to see the changes in ammonia in green, nitrogen in nitrogen in orange, and phosphorus in blue. And so you can see when I add those rains back, you can see exactly where the peaks of each of these were, and especially this really cool finding in that same window of time, probably during application. Now, despite that, Gabe, despite that, our title is still true.
Yes.
Even though we want to argue with our title. And that is shown in this graph, if you want to unpack this for us.
Yeah, this is actually this is probably my favorite figure in the presentation today. Here we're showing the proportion of the pounds of each of the nutrients, phosphorus, nitrogen, and ammonia, that came in what we are calling our spring totals versus the rest of the year. And what that simply means in our spring totals, we're looking at April and May, and we're separating that out from June to the end of October. So you would expect, you know, if the blue proportions of these columns are just 2 months of our entire sampling season. So especially with phosphorus, you know, we're looking at pushing 80% of the total amount of phosphorus that came down the Maumee River last year came in a 2-month window, right? So that's significant when we're trying to, with these common goals of reducing the amount of phosphorus coming to Lake Erie. I know a major goal was to reduce by 40% by 2024. So when we're trying to think of how do we move the needle, how do we bring down the total amount of phosphorus that's going to Lake Erie, I think an incredibly important piece of information is to recognize that here in the Maumee, 80% of what came down last year came in a 2-month window. So if we can utilize conservation practices that are targeted towards the spring rain events, and also that address the things that are happening on the landscape during those windows, I think that really points us in the direction of how do we get that 40% reduction of phosphorus to Lake Erie.
Yeah, it's the low-hanging fruit. Absolutely, the first of the spring. And, you know, even that spike in July, remember, those spikes in July are up here. So, you know, the spikes in July are probably 80 or 90% of what's at the top. Yeah. And so I think that's part of, to explain our ammonia data here, because if it's, you know, farms, dairy farms or others, you know, applying chicken manure or dairy cow manure, that can explain why the ammonia, remember, these are not amounts, these are percentages of total. Okay, so Jennifer, any questions that have come up? Yeah, fire away.
So I have one that's specific to the slides that you're showing. Could you just go back, or you don't need to go back, but remind me, is this all of this data from the Upper Maumee site, just that one site? And then are you going to talk about the other 2 sites, or do you have a comparison?
Yes.
Okay.
No,
so we don't. So that question can wait.
Well, we knew that the Maumee would be the most important. Remember, we've got all these other sites. I mean, all the brown dots. Gabe's team is now crunching all the way back to 2022 and maybe back to 18. So, I mean, you know, Gabe's got a new thesis project if we can. But from 2024, we have the other roughly 19 sites. I think we were at 22, 23 sites this year.
Per year, yeah.
Yeah.
That's about where our campus is.
So, and we're making decisions now about what sites we'll use this year.
Yeah.
Obviously, we want to look at all of them. We are not here, and I'm looking at people online, we are not here to point fingers. That is not the point at all. Our hope is that, my personal hope is that we can each find something to address. So developers, you know, with all the runoff and the total suspended solids, there's a lot of development in Fort Wayne. Maybe that's a cause for TSS spiking in the early spring during early thaws and development. You know, as we mentioned, lawn care, golf courses, all of these, if there's a way we could get in front of that, and then of course farmers and their application practices and fertilizer practices and getting them to do more soil testing to know for nutrients. All of these are the projects that we're hoping to go after. But the data we've just shown you has to be redone for 23 sites just from 2024. So we have those, we have them all done, but we're not going to bore you with them all. I mentioned if you wanted to go get a coffee, I hope you're back now. But we will go, you can close your eyes. I'm not able to forward a whole bunch at a time, but we're going to go to that next spot. So we'll travel north. Whoops, let's get over here. So the spot we were looking at was right here, the Upper Maumee, that blue dot right there. And now we're going straight north. What do you think, Gabe? 20 miles? 30 miles?
Maybe a little more. Yeah.
Yeah, because you can see some of the counties here. So 30 miles probably up close to the Michigan line. And this is going to be our St. Joe, Ohio line.
Right. Yeah, we're catching, if you're familiar with the town. Oh, you had a question?
Yeah, just a follow-up question. Can you also then just remind us, are these the data points that you're collecting? You showed us your manual collection method, or any of these with the USGS automated sites? Just real quick, you don't have to go into the detail. But.
Yep, everything we're showing you today is based off of our manual collection system.
Yeah,
yep.
All the catch sampling. That's why this is so remarkable. And those, you know, there are foibles with everything. There's breakdowns. There's continuous data on some of them, but there are not very many of those continuous monitors. And so we wanted to really compare the spots that we thought would matter. You know, Ohio wants to know what they're getting from Indiana, and Indiana wants to know what they're getting from Michigan. And, you know, again, no finger pointing, just information.
Yeah. Thank you. And there's a really good question about grant opportunity.
Go. Yeah.
But I think we'll save that for the end, because that's kind of a more summative.
Great.
Yeah.
Okay.
So we're going to be simple here. We're just going to cut to the chase and show two graphs. So the first one is the total suspended solids from that St. Joe site. And the reason we wanted to show this is you'll see all the nutrients in a moment, because we've got the compiled graph that we'll show you. But first of all, the rain events are slightly different on the St. Joe. Like we mentioned, we're further north. It's a different weather pattern coming out of Michigan. So you see the rains, while the heavy rains are kind of in the same amounts, they are more frequent, if you will, and higher across the board in the St. Joe. And then we see this cool coordination with this. So we're at total suspended solids versus precipitation, and those precipitation events are here, so you can see them on the next graph. But if you want to summarize here, Gabe.
Well, I think the main thing I would just point out here is we're seeing the same pattern that we saw in the Maumee River, right? So we're seeing the area under the curve during April and May here is much greater than what follows. But same pattern, we're seeing the spike in July. So now we're going to move, and we're just going to summarize all three nutrients on the same graph.
And so just like we looked at previously, on the Maumee, we're looking at the St. Joe, the Ohio St. Joe, right? Coming in from the north part of the northwestern Ohio. We're looking at rain events, those same rain events shown here with the blue arrows at the top. And we've got phosphorus in blue, nitrogen in orange, and ammonia in green. And so to Gabe's point, we had to show this again, like we mentioned before, on a log scale, because the ranges are so different. So the peaks don't look quite as peaky, as spiky, but that's because that's a tenfold difference between every line. And so I think you can see the point, except with the ammonia. The ammonia does continue to spike and then finally reaches its lowest spot. And I guess they all do that, don't they, Gabe?
Mhm.
Phosphorus would be the only one that comes down pretty low in the late spring.
Yeah. So.
Yeah, that, can you speak to that?
Which
anomaly there, that
Yeah.
phosphorus that comes down?
And it's just, there was a dry spot, there was a dry time, and we know that's when farmers were applying. So it was dry just long enough for them to get out in the fields and get the work done. Yeah. So that was where it came down, and then we had another big rain, and then the one right at the beginning of the
Yeah.
July.
Okay.
So, not certain. And I would argue that it was maybe a problem with the assay, right? The testing, except everything else went up. So it's not like the sample was bad. And it did only go down as low as some of our lowest points, which if the testing was completely wrong, I would have expected it to go down below 1. Yeah.
Thanks.
But that's just, I am with you. Crazy.
Okay.
And
So
so, yeah.
we just covered that. So
Yeah.
now this is my favorite slide. It's my favorite one to go into. The St. Joe is a little more, what's the word? Balanced, but still you're seeing in these blue columns for the proportion wise for the total amount of the nutrients that came through down the St. Joe River as it was coming into the state of Indiana. We are seeing that over 60% of phosphorus and nitrogen came in a 2-month window. So that's really the story. Where with the Maumee, we were looking at sort of a 80-20 difference. Here in the St. Joe, we're looking at a 60-40 proportion-wise. So finally, we want to move on to the St. Mary's River. This is down near Wilshire, Ohio, as the St. Mary's River is coming into the state of Indiana. If we saw the continuation of this map, you'd see that it goes down at the headwaters near Grand Lake, St. Mary's, if you're familiar with that, and it comes towards Fort Wayne. So as we dive in, we are looking at all 3 nutrients. We're going straight to the summary here, and we want to highlight again that we have that same response early to mid-July here.
And that response, Gabe, I mean, if I look at the magnitude, granted, it's logarithms, which tells me those peaks on the St. Mary's are much higher than I think the peaks we saw previously. Yes. And we would only get to that number, as you students know, by conducting these tests, the statistical tests that Gabe was talking about, and doing areas under the curve. But it is staggering how much of all 3 nutrients do come back. They do rebound to almost spring levels here. Not quite. They're still about tenfold lower.
Yes. Yep. And then again, with my favorite slide, this one, we're looking at an 85-15 sort of, almost a 90-10 relationship between April and May compared to the rest of the year. So that definitely catches our attention. Just if you were to prioritize, where do you want to direct your efforts, just from that perspective.
Right.
Right.
So those are our data. We didn't want to go into too much depth on the second 2 sites, the blue and the red here. We spent most of our time on the Upper Maumee. And so what Gabe did then to bring this full circle, and this might answer some of burning questions, is that he then decided, okay, the Upper Maumee might be just a larger landmass, or maybe the St. Mary's is a larger landmass. Maybe this looking at, pounds per day isn't fair. Let's normalize it, if you will, by acreage. And so if we take the number of pounds per day and we divide that in by acres. Gabe's got the next comparison.
Acres of the watershed. So on these bar graphs, we kept with our color coding system. The Maumee Watershed is in yellow, the St. Mary's Watershed is in red, and the St. Joseph Watershed is in blue. And like Julie was saying, to sort of cut out the differentiate, because certainly when we're looking at the St. Mary's and St. Joe coming into the state of Indiana, we're talking about a much smaller drainage area than what's combined in the Maumee leaving the state of Indiana coming into Ohio. So we divided by the amount of acreage in each watershed to really just break down how many pounds of each nutrient are coming from each acre in that watershed. And you see those proportions as a percentage here if you were to break everything out. So it shows you sort of a balance. And it makes sense that the Maumee, which is shown in yellow, is really simply an average. I mean, it's literally the red and the blue coming together. So it makes sense that what we're seeing in the Maumee, that proportion is roughly 50%, or sorry, the average between the two of its contributions, the St. Joseph and St. Mary's rivers. So is there any other points you wanted to?
I think this is powerful, and I think this is where our strategic planning people should be looking, right? Our counties and our soil and water conservation districts and the NRCS, right? These are the data that I think we could use to really make arguments of where to make changes. And then we could see incremental changes. So that's our goal, is if we could have this graph since 2018 and see what those percentages are, maybe things are getting better. I think that's the hardest thing for our farmers in particular, is we keep saying, join this program, do these conservation practices, and they're like, okay, my yield's fine, what's it doing for you? You know, this is a lot of work. And so I think that's where we wanna.
And you're hitting on it. What are the next steps?
Yep.
Where do we want to go from here? And that would certainly be in our minds, that would be the next most important thing to see, and exactly what we're already looking towards is building out their data set into the treasury of historic data that has been handed on to us. And of course, if by every means, if we can continue that sampling as well, not just to look backwards and be able to get that a pulse on what direction are we moving in, right? Because that vision breeds excitement or enthusiasm towards accomplishing the goal that you're setting before yourself. By knowing where we're at and how we've been doing can really inform and build interest among landowners and interested parties to sort of put wind back in their sails. So that's really kind of what we'd like. And that's that education, outreach, and collaborate portion of this slide. Yeah.
So you're, maybe you read the question in the chat, but the question here really goes to your first point about continuing sampling. So I'll just read this. Doug Kane, Dr. Kane, who's a professor at Heidelberg and was previously here at Defiance College, he says, have the various groups ever thought about writing a grant or finding another source of funding to start automated sampling at a few sites? Maybe those 3 state line sites on the St. Joe, the St. Mary's, and the Upper Maumee.
So Gabe can brag now, and he can brag on Greg Lake also, because these sorts of things are being done by the USGS, the United States Geological Survey, and we're involved. Gabe is really involved in a new project in Eastern Allen County.
The new project that she's I think you're referring to, is this 2-stage ditch project. So at a property there, Greg Lakes, right now we're in the preliminary stage where we're gathering the baseline data before the installation of the 2-stage ditch. So now we're getting a pulse of how things have been, and then next year, towards the mid to end of the year, the ditch will be installed, and then there will be a period afterwards that will monitor that, and using those automatic samplers. And certainly, even to this water route, it certainly crossed my mind. I actually reached out to Goshen College last summer, because they have sort of developed a sort of automated system for the size of streams that we are looking at. They're doing a very similar project, different watershed, all that's Mississippi. And I looked at their number, like, so they shared some information with me, and it would just require a significant collaboration financially.
And by significant, we're talking 5 to 6 zeros. So these can cost hundreds of thousands of dollars. And in the case of the 2-stage ditch, the way that they've done that is they're sampling upstream of the ditch and downstream of the ditch to see the differences in water quality and how the ditch makes a difference. These are hard. These are hard and they're expensive. And I think we've all been watching the news, so we know what's happening with funding. So that's our issue.
Well, and I think you're going to an important point that there's the tributary sampling, right? The main stem of the Maumee River, or the St. Joe or the St. Mary's River, but then there's also the monitoring that's happening on the BMPs or on in the field. Right?
Correct.
That are, like you said, upstream and downstream of a practice.
Correct.
Which is a very different data set. And how effective is something if we install this?
Yes.
If we install, multiple overwide ditches for a low investment, how does that compare to, $1 million wetland, for example? That's a question that's been asked.
Exactly.
So we don't know unless we're monitoring those practices.
Right.
Well.
So if anybody has, 50 acres that we could put into a wetland right upstream of one of our sampling, we would love it. We'd work with Acres or some other land trust. That would be beautiful.
Yeah, because what you kind of hit on, what we talked about today is our most macro view of what we're doing. As you go through the rest of our sites that we visit, the other 17, there is a few more baseline sites that we have, but then we have about 5 where each year we can kind of go and look on a more micro scale, the sub-watersheds, to see what's going on. And usually there's either something going on in the watershed that's of interest, where we want to see, if we anticipate something's coming, we want to catch the before and after of it, something like that. So, but then, so we can drill in and out. This was the most wide view that we can do.
And before we leave this slide, I just want to say this is a picture from the Defiance College website, and these are Defiance College students. And Jen, I don't know if you're in this picture or if any of the other people online are here, but this shows indeed another form of water sampling that you can do either in our state. It's called the Hoosier River Watch. I think you all have something similar, and I've seen other grassroots groups as well as colleges. I know Heidelberg has just some phenomenal data. So I'd love to talk more with you, Dr. Kane. But we'll just skip our last idea and get us to the thank you then. Defiance River or Research Alliance, Upper Maumee Watershed Partnership, and, your soil and water, our soil and water, and all our neighbors in between. We're ecstatic. So we'd love to answer your questions. We'll go back to slides you want to look at. I know we're within about 5 minutes, so I don't want to keep anyone too long.
We don't have any more questions in the chat right now. If anybody does have another question and they would prefer to just voice their question, I think that's fine too. I think everybody has the ability to unmute, so I think maybe raise your hand or just jump right in if you have a question. Kat, do you have questions? No. Well, I think we can give everybody a few minutes back of their hour. That's okay.
Exactly.
But we will have a recording of this available. It'll be going out. All of you that signed up will be getting an email with the recording. If I'm coming through okay, I am going to direct you to Dana Peterson, and I think you all got an email from her with the link for this meeting. So Dana will make sure that you have access to the recording. And then I think the last thing we want to point out is that we're going to be having this conversation again in the beginning of April. April 2nd and 3rd, we're having a conference here at Defiance College. We've got the first day is April 2nd, and it's going to have a lot of focus on student engagement, science, and kind of the future and conservation for younger participants. And then April 3rd will be more of a watershed style meeting with presentations and keynote speakers. So keep an eye on your email for the information on that. Julie and Gabe, thank you very much on behalf of Defiance College, the city, everybody here in our Upper Maumee Watershed, thank you for sharing with us.
You're most welcome. And thank you so much for inviting us.
Absolutely.
You really are some of the first people just to hear this presentation. So you're really on the cutting edge. So thank you so much for giving us the opportunity to get the word out.
This was great. We got lots of very interesting, great, thank you very much. So we will, Dr. Kane, we'll follow up with your question because I think there's some opportunity there.
Absolutely.
We don't know the funding, scenario going forward, but we know what work needs to be done. So thank you everyone for joining.
And we'll stick around if anybody wants to ask more questions.
Okay, sounds great. Thank you all.
So
We're good.
I don't know how to, I couldn't screen, how do you share?